All right. So for the lab, um, last class I talked about more of the details around independent, dependent variables and the pre-lab and this and that. Today's lab is more one of these demonstration labs. We're going to be doing, normally you would do it, but instead you're going to watch videos of, me and Tina, like the other instructor, doing this stuff. But you still are going to be able to see in a more physical way these different concepts we've been talking about be demonstrated in ways that are hopefully going to make it a little more real to you rather than it's like, oh, all these words, hydrophobic, hydrophilic, dissolve, doesn't dissolve, conducts electricity, blah, blah, blah. Um, you'll get to see things, you'll get to hear things, you'll get to write down your observations. So there's no hypotheses. However, you still do need to analyze the data. You have to interpret, you have to talk about what it means. So, like when I'm grading these, let me go to, let me go here. So your lab write-up. So a few things I should mention. So one, it is in your lab notebook. So this is not something that you do on scratch paper and then make some pretty perfect version for the end to turn in. Um, a real lab notebook is kind of a record of your observations and things like that. It's not some beautifully formatted thing. Um, so while you're in, it also means that like when you're done with the lab, that you don't have to, you're kind of got it's down already. You don't have to go back and do something else. So it should be in your lab notebook, should be in pen. The idea here is that you're not changing things or making things up. So if you decide like, oh, this is not what I want, like I made a mistake, or it's going to happen, I promise you, you're going to have some objective or hypothesis, and I'll give you feedback and say, actually, it's got to be modified in this way or that way. You never erase. So no erasing. Cross stuff out. uh, if you need to. So you might have actually somebody. We need to turn off somebody's got some feedback here. So you might be writing like objective blah blah blue de doo de doo, and then you realize like okay, I totally missed something there. So you might end up just putting a line. Don't obliterate it, and then write in a revised objective. But it should be clear that there was this process, right? And you don't lose points. You actually are going to lose points if you are actually obliterating things or erasing things or whiting things out. Or that's not what this should be. It should be kind of a record of your process. And I know, particularly in school, where people start getting into this weird, like, it kind of trained, like, what is expected? Better make it look perfect. So, like, this is really supposed to be a kind of a record of your observations. It's supposed to be a place where you analyze things, um, but it's not supposed to be beautiful. And people get really, really stressed, thinking that I'm going to see that you're not perfect, that oh my God, you wrote an objective and then you had to revise it. It's like, I'm, that's not shameful. That's the process of science. That's normal. That's normal. So I know as much as I say it, people get stuck in this idea of feeling vulnerable when somebody else sees that you're not perfect. But the lab notebook is graded on ultimately, do you have a good objective? Do you have the data presented well? Do you analyze? Do you kind of talk about what it means in a good way? But it should all be handwritten. This is not something that you compose on a computer and print out or something. This is something you're going to write in your lab notebook. You'll do the scanning with the little scanning app on your phone and just submit it into the Canvas shell.
I have a question.
Uh-huh.
Should we leave space for a possible revised objective, or like can we add that somewhere else later?
So so what I would say in reality, ideally, in an ideal world, you never leave blank space. You just kind of keep moving. Sometimes you might find if you're taking some data and there's going to be a certain little discussion and you're moving on, you might want to leave some space for yourself. But even when you do that, sometimes people run out of space, in which case people do this. Remember, each page is numbered. Like let's say you're on page 14 and you've gone la la di la di doo di doo di doo di doo. And now you've gone on to the next lab on page 15 and it's like some new lab. And then you realize, like, oh my God, as I'm going back here, I'm realizing I need to redo this whole section here, or I've missed out talking about some important thing. You can just put like, additional, discussion on page, 40. And then later, on page 40 here, you can put a big star and continue discussion for chemistry lab or something. So if you do run out of space, it's not a disaster. All you need to do is, have a clear little link to where it's continued and then write it someplace where there's room.
Thank you.
It also means sometimes people get kind of clever, like pasting things that flap open, and there's this here and there's something. Don't do that. Everything should be flat. Everything should just be flat. You should be able to take a picture from page to page and see everything you need to see. Um, the things you will be pasting in, you'll be pasting in the methods, which are just in the lab book. You can just cut them out and paste them in using rubber cement or tape or glue stick or whatever you want. Um, Graphs and things that you print out from the data, those are things you can paste in there as well. Um,
I also have a question. So as far as like what we go over in the Zoom class with you during lab, should we be like putting everything, all our notes in there right away, or after we do the lab, we then put it all together?
so neither of those.
Oh, okay.
All right. So good. So let's talk about the lab book in a little more. So lab notebook. So far all I've really told you is it's got to have a table of contents. So hopefully everybody's got page 1 as a table of contents already. And the first lab today is going to be this chemistry review lab, which is probably going to just start on page 3. It's a kind of a good place for it to start. So in your lab notebook, there's always going to be kind of some objective, like what is the lab about? If it is one of these labs that I've highlighted in fuchsia, right? The ones that are hypothesis driven, you're also going to need, you also need like the hypothesis and sources of variability, if appropriate. So there's kind of like, what is the point of this thing? And again, that's usually coming out of just in the lab manual. Then you're going to do your methods and materials. This is typically just cut and paste from the lab manual. So let me find these things I got from last time. Hold on a sec. Lab, 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 lab. Simple labs. Here, so I'm going to share a screen. This is somebody's lab. This is the jumping lab, the next lab. And she has her name. That's the other thing. You're going to have your name at the top of each page. It makes it easier for me to know that it's your lab. And I can also see the name and the numbers and make sure it's all kind of happening sequentially and all that. But, she's got an objective and a hypothesis, sources of variability, because this one actually does have a hypothesis driven. On Tuesday, we're actually going to talk more in detail about the expectations for your jumping lab hypotheses, but we'll talk about that more then. But then look, for the methods, she just has a cut and paste from the lab manual. That's why everything is done single-sided. I printed all of the methods single-sided in the lab manual. Then she has her data and results. Most of this is going to be stuff that you're kind of recording as you're doing stuff. Although, because you're not actually doing stuff, you're actually going to be getting data that I'm going to give you in spreadsheets and stuff like that. So this could be where you have data that you put in, or it could be observations. Like today, you're going to be writing observations of what you see on these YouTube videos as we do the different parts of the experiment. So the data and results and it's supposed to be a real record of what you're seeing. And you want to describe it in a kind of complete way. So if you look at it 2 months from now, you'll be able to oh yeah, that's what it looked like. Don't just say yes, no, or something. Kind of describe things. I'll talk about this in more detail. And then the most important part is going to be the discussion. The discussions are where you actually show that you actually understand how your observations and the data fit into the bigger concepts of the class. This is where you're going to have graphs. A lot of the graphs, some of them you'll draw, a lot of them you'll be making in Excel. All graphs always have to have a graph number that then refers to things in your discussion. And then you're going to have your discussion where you kind of describe what happened, what you found, and how it fits into the big picture, how it addresses your hypotheses if appropriate. And here's another one. So this one, she has actually, this is actually worth putting in here. Notice that she put this background information here, this up at the top here. Some of the labs have background information. The chemistry lab has background information written in the lab manual. It is okay to put that in your lab notebook. And it probably makes sense to, because you are allowed to use your lab notebook as a resource during the exam. So if you can have extra material in there, why not? Then she's got the methods and materials here. Here she's got her objective hypothesis, data, here's her graphs and her discussion. So just kind of putting this all out there that it's kind of like the objective, maybe some background, which is coming out of the lab notebook. There's going to be the methods, which again are coming out just straight out of your lab notebook. Just like I tell students, it's one of the few classes beyond like kindergarten where you really need to have scissors and paste to do well in the class, you know. So this is just going to, methods are just cut from your, you know, because it seems silly to write them, rewrite everything. So cut from the manual. And if we do change things, sometimes we'll change things up a bit, then you'll have to note that as well. Then there's your data. So these are your observations. This is important that you write down what you're seeing and not what you think you should see. You know, because, and also sometimes the experiments don't give you the answers that you think they should. And that has to be all right. You have to write down what you actually notice rather than what you think, you know, what does the world look like rather than what do you think the world should look like. That being said, in reality, if you get some observations that are really off from what you expect, you know, it's worth double checking. Like, did I do something totally wrong? Did I spaz here? But in general, the observations are what you see. You know, again, for our lab today, you're going to watch Tina and I mixing things in different solutions, and we'll show you on the screen and describe them using words. And in your lab notebook, you are going to write down in longhand, you know, for this part or that part, what is happening here and what are the observations. So this is minimal interpretation at this time. This is not about what does it all mean. This just means like, what do I see? Then comes the discussion. This is, what does it mean? So this is where you have to take the observations and describe what the significance of them are, how they tie into the concepts of our class. You know, for our lab today, it's going to have to do, we're going to have polar solutes, nonpolar solutes, polar solvents, nonpolar solvents. We're going to mix them up. We're going to try putting electric currents through them. We're going to do all sorts of stuff. You're going to get all this data that what happens as you mix and match all these different things. But then in your discussion, you're going to have to bring it all together and talk about the bigger picture. Like, what is this telling us? Why does this make sense? How is this illustrating the basic concepts that I was talking about in lecture this morning? This is where a lot of your points come from, right? It's one thing to be able to write, you know, sugar dissolved. It's a very different thing being able to say, like, why the sugar dissolved, why it made sense, and why the sugar dissolving is different than the salt dissolving, how it's similar and different, right? So the discussion is where you're going to have all that kind of stuff. You know, the data is more just kind of describing the observations. Discussion is where you get deeper into what it all means. So back to the earlier question, the lab notebook is not a place where you're going to kind of put crib notes or the notes that I talk about in lab. Instead, it is going to be a place where you have, this is what our lab is about, this is how we're doing it, this is what we see, this is what it means. And it shouldn't have like little crib notes or things in the side or something like that. It should just really be about the process of the science. That being said, when we discuss, like, in fact, I'll always be doing wrap ups and talk about, what are the main things you should make sure you address in your discussion? What are the main take home messages here? So you should pay attention to that and make sure that that finds its way into your discussion. Your discussion should address the main important messages that I am kind of yapping about in our kind of lab kind of lectures. But you shouldn't be taking like lecture notes in here. So not lecture notes. It's this more formalized thing, the objective, the methods, the data, and the discussion. But again, these lecture notes are going to help inform you. Like, I'm going to be talking about how to formulate your objective and your hypotheses next class for the jumping lab. We'll be talking about take-home messages for your discussion at the end of today's lab. So, any questions about the kind of expectation here? And again, this is all written in your notebook. In pen.
I have a question. Can we use different colored pens in the notebook or just black?
You can do different colored pens if it's useful. Don't do some like super light color that's going to be hard to read or something. Obviously. Yeah, and in fact, it might end up being helpful, like doing some graphs and things like that. You might find that particularly helpful. Other questions?
Are we handing in our labs after every lab or just kind of when you ask us to periodically?
So for like this lab, in fact, let me take a look where I put it. I actually, give me 1 second here. Modules. It's usually I give you 2 weeks after we're done with the lab to actually submit it.
Okay.
Although for the objectives and hypotheses, which you need it before we start the lab, those will discuss in the previous lab. So like next class, we're going to talk about the expectations for the jumping lab objective and hypothesis, and that will be due on in time for the jumping lab, so that it'll be due the next class. But that's a short little thing. That's like a quarter of a page.
Okay, so that's going to be handed into you or just expected to be done by the lab time?
It'll be handed in.
Okay.
I want to make sure it looks good. I have separate links to turn those in.
Okay.
So we'll have
Thank you.
those, but then for the jumping lab, the main jumping lab write-up won't be due for 2 weeks after we're finished with the jumping lab. So that's the basic way I kind of did it. I kind of giving you enough time that you can get to it, balance other things in your life, ask questions if you're running into trouble, and at the same time making sure you don't just leave it till the too long, and then because people who wait too long, they just end up buried. And last semester there was one person who, trying to think if she either went down a whole grade or failed ultimately because the lab notebook ultimately wasn't done. Just kept not getting to it and not, actually it wasn't like this person didn't even really understand the material, but there is something about the process. You got to actually get it done and turn it in and show that you've thought about these ideas and kind of written this up.
Okay, thanks.
Cool. All right. So
I do have one more question. Sorry. Are you going to tell us what page this lab should start on?
wherever is free next. right? So
Okay,
we're
so,
on this page that's free.
okay, but this lab should start on page 3 because page 1 is our table of contents, right?
Check. That sounds about right. You know, and
Speaking of page numbers, I was looking in the lab manual as well, and it doesn't look like. Oh, never mind. Never mind. I just figured out that one answer to my question.
okay. All right. You know, and as you're doing this, if you have questions, just ask me. You know, I'm assuming that there's going to be hiccups because, you know, this is a new thing. As we continue doing this through the course, you'll get more familiar with the expectations. But if you're writing things up and you're like, am I on the right track here? Like the students that did best are the ones that would like, you know, email me stuff in between and say like, does this look good? And I'm like, yeah, no, that looks good. Keep it up. Or like, oh, actually, you might want to like, you know, kind of adjust this or that. You know, so I am happy to give you feedback. Or in the office hours, that's another time where people go over their labs with me. Again, once our class is done, you can stick around. I'll be here for an hour after our official class is over to discuss anything you want to discuss. Um, good. All right, so chemistry review lab. Let's go. Where is Doobie-doo? There it is. A few more moments. Somebody has gotten noise. If someone could mute there, I think that's it. All right, I'm going to share screen. Okay. You know, the chemistry review lab. Again, here, this top part here is just basically the objective. This one here is not a hypothesis driven. You know, it's basically saying, you know, this lab is a number of exercises that demonstrate and reinforce the chemistry concepts important in this class. While they do not require a formal objective and hypothesis, you must still record your observations as well as summarize the significance of what you find. So that's that. And I realized because we haven't actually talked about pH and we haven't talked about chemical equilibriums and stuff yet, probably makes sense to start on part 2 today. We'll do like parts 2 and 3 right now, which kind of go together. And then we'll do part 1 on Tuesday after we've discussed pH and chemical equilibrium in more detail. So that way we'll kind of reinforce the stuff we've just been talking about, and next class we'll reinforce the newer stuff we've just learned about. So in your lab notebook, what you probably want, you know, we're going to start with part 2, you know, which is basically electrolytes. And this is actually, this little picture here is useful to look at. You know, here's a salt crystal. Here's a salt crystal at a molecular level or an atomic level, like the minuses are being the chloride ions, the pluses are the cations, the sodium ions. But if it's just as a crystal, they're in this crystal lattice. But when you put it in water, all of a sudden the anions, the cations, they're all independent, just floating around like this. It's actually, I can. This is actually this. This is like when this thing Eric let me grab. Hold on, let me get past. Okay, so a solution is basically, you know, these little Mickey Mouses are water molecules, right? The red is the oxygen, the white is the hydrogen, so H2O. Each of these Mickey Mouses is one of these little bent polar water molecules. These purple ovals are supposed to be some solute, some stuff that you're putting in there to dissolve. Right? So this is kind of at a molecular level what a solution looks like. Again, solvent, just by definition, is a thing where there's more of. Solute is the thing that's kind of less of, that's kind of being dissolved in there. We'll go over this later. We are going to talk about colloids, but let's talk about dissolving salt. So here, again, when you first put the sodium chloride in the water, it's a crystal. And the sodiums are pulling on the chlorines, and the chlorines are pulling on the sodiums. But all of a sudden, there's a whole new game in town. There are these polar water molecules, and these guys are off for adventure, and they're going to go dance with all these new characters floating around. And this starts dissociating. And you get individual sodiums, individual chlorines start finding their own adventure, floating around, dancing amongst the water molecules. Again, this is dissociation. Not only is it dissolving, going into solution, but it's breaking apart into individual ions. Right? So, and again, the thing that's interesting is for every one NaCl, there's now 2 ions, 2 particles. So it's actually going to have major implications on osmosis when we talk about that in more detail next class. Also, again, electrical current. Electrical current is just flow of charge. And all of a sudden, we've got free charge that can float around. That means that we can have electrical currents flowing through this solution because we have free charge floating around. You know, solubility equilibrium. We'll talk about equilibriums and more, but eventually, depending on how much salt is there, it's either all going to go in or some will not. Again, electrolytes, pure water. We're going to see there's a little bit of conductance, but very, very tiny. Salt, though, is going to conduct electricity very well because the electrical charges can move from one electrode to the other. Will sugar conduct electricity?
I don't think so.
Why? What is your reasoning there?
I think sugar is a nonpolar molecule.
So sugar
Because
Or-
it
dissolve if
doesn't
it's not polar.
dissociate.
Say it again.
Because it doesn't dissociate.
Exactly. It doesn't dissociate. It's most definitely polar. It wouldn't dissolve if it wasn't polar. But it goes into solution, but it doesn't break apart into individual ions. It stays intact as an individual sugar molecule, which overall is neutral. It has charge imbalance, but it does not have like a pure charge, like a plus or a minus, like a sodium or a chlorine does. So sugar does dissolve. So salt and sugar are similar in that they both dissolve. They'll both go into solution and disappear. But the salt is going to conduct electricity because you have individual charge carriers from the dissociation, where sugar, it's not, right? Because there's no charge carriers. You know, HCl also dissociates into H plus and Cl minus. So this is kind of showing this difference here. When you dissolve an electrolyte, you know, the salt, NaCl, breaks apart. We have individual pluses, individual minuses, anions, cations in solution free, doing their thing. When you dissolve like sugar, it goes into solution because the charge imbalance on the sugars will do that little electrostatic dance with the surrounding water molecules, do hydrogen bonds, but they don't break apart. They still stay intact as individual overall neutral things. So these two pictures should make sense. Does this make sense? This is just kind of reinforcing what I've been talking about in lecture. All right, so just kind of, again, trying to review this. And again, hopefully none of this is brand new. This should all feel like, oh yeah, let's dust this off. But, and like I said, it's at the core of so much of our class, trying to understand polar, nonpolar, hydrophobic, hydrophilic, dissociation versus just dissolving. Dissolve but not dissociate, dissolve and dissociate. And the basic idea here is you're going to take, you're going to watch us take a little bit of sodium chloride, potassium chloride, calcium chloride, add them to little beakers, take a look at it. And here it says, describe the appearance of each of the dry electrolytes. What that means is you are going to probably have to pause. I'm going to go over this with you, but then you can still look at these videos on your own. And you're going to want to write down in your lab notebook, longhand, you're going to write down, all right, part 2, 1A. All right, sodium chloride, describe what it looks like. It's not going to be a shock. Sodium chloride is table salt. Looks kind of like white little crystals, right? But you have to write that out longhand. What does it actually look like? Then potassium chloride, what does it look like? Calcium chloride, what does it look like? And again, you're going to be able to see from our video. Then it says, then add 5 mils of distilled water to each beaker. Swirl the beaker to mix the contents. And then record the appearance of each beaker and compare the results. So now you're going to record the data when it's been dissolved. And again, this is going to be in the video. Then test each solution with the electrical continuity tester. You'll see in the video, but basically it's a buzzer I've made where if the 2 wires touch, it buzzes. And if the wires are not touching, it doesn't buzz. And if you put the 2 wires in a solution, if the solution conducts electricity, you'll know because you'll hear a buzz. So it's a very just, brute force way to see, does this thing conduct electricity? It's basically 2 wires connected to a battery and a buzzer. You'll see it in a few moments. It says for each electrolyte, so for each of them, you're going to say whether it buzzes or not. Then it says you also want to write the equation that represents the dissociation in water. For instance, potassium bromide becomes potassium ion aqueous plus bromine aqueous. And then it says choose one of the above electrolytes and add a pinch of that electrolyte to oil. How is trying to dissolve something in oil different from dissolving it in water?
Oil is nonpolar.
Exactly. This is going to be putting our salt in a nonpolar solvent and see what happens. And then describe what it looks like. Describe the results. Also test with the electrical continuity. See if it conducts electricity. So that is part 2. Part 3 is going to be the exact same thing, except instead of using salt, we're going to use carbohydrate. We're going to use sugar. Basically, you're going to add a pinch of glucose to the beaker and describe it. Then you're going to add water and see if it dissolves or not. Test it with the electrical continuity. See if it conducts electricity. And then this last part here, it says compare the jar of glucose solution to the jar containing starch. What do you see? Describe how they look. So again, we're going to do this. You'll see it. And what is the difference? I have to mention this a little bit to you. But you're going to describe this. Again, in your lab notebook, part 3, part 2, describe what you see. This looks like this. This looks like that. It says, draw the structure of glucose in the ring form and show at least 2 water molecules attracted to it. And then it says, be sure your write-up for this has a discussion and conclusion that summarizes, compares, and contrasts the results from testing different types of compounds. Like in your discussion, you're definitely going to want to be comparing how the sugars and the salts were similar and different, and why. This is where you're going to be able to bring in all the theory that we're talking about of dissociating versus just dissolving, and why did it dissolve? How are they similar? How are they different? So the first part is your observations, which you can do now, but then you're going to have to do a more elaborate kind of unpacking of what it all means. So are there questions about the core idea of what we're doing here?
I have a question. So we write all of that down, and then there's 3 pages for this lab. Do we post all those 3 pages in our lab book?
Let's say it again.
There's 3 pages of the chemistry review write-up. Are we then taking all those pages and putting them in here? All 3 of them?
Right. So I mean, I would do it a little more, what do you call it? Thoughtfully here. Like, at the very beginning of the lab, I would just probably have the title and this little initial part, which basically gives you the, the description of what the overall lab is. Then I would probably cut out, the instructions for part 2, because that's where we're going to start for today. So the methods for part 2 here, you'd have part 2 electrolytes, and cut it off there, and then take your data for this part. Again, your raw data. Then paste in the instructions, the procedure for part 3, and record your data from here. And then you'd write kind of a more elaborated discussion that brings in all that data and says what it means. So it will ultimately be in there, but I think it'll have a much better flow if you have the methods and then put the data after the methods. Also, I would say make sure you don't lose part 1, because we are going to be using that one next week. Hold on to the buffer systems. I wouldn't paste it in there yet because we're not going to that part yet, but I would make sure you don't lose it. Have a folder or a binder or something where you're making sure you don't lose the little slips of paper.
So for today, you want just part 2, part 3, and you still want a discussion based off of those 2 parts, or does the discussion come after all 3 parts?
I would say the discussion will have 2 kind of set. Parts 2 and 3 have a common discussion, and parts 1 has more of its own discussion.
Okay, so it'll be 2 separate discussions.
Yeah.
Okay.
So let me, in order for that part about starch versus glucose to make sense, I need to bring one more little point in here. So I talked, in that picture we said there was a solvent, which is the stuff that's kind of in the majority. There's the solute, which is the stuff in less quantity that is kind of dissolved in the solvent. So this is my mixture here. I have the solute in the solvent. There are 3 different versions of these mixtures. And I'm just going to briefly talk about that just because it's going to make part of this lab make more sense. It can be a true solution. So when we talk about a true solution, this is what you get when you're dissolving sugar in water or salt in water. The hallmarks of a true solution: solute never settles and does not scatter light. Which means it's transparent. It might have a color, but then it looks like colored glass.
I'm sorry, what did you say? It does not scatter what? Because it's kind of cut off on the side.
Light.
Okay.
So it does not scatter light. Um, 2. Another possibility is what's called a colloid. So solute never settles. So that means once it's gone into solution, it stays there, right? Either a true solution or a colloid. You can leave it on the shelf for a week and they're still mixed up when you come back. However, it does scatter light. It ends up looking kind of milky. The basic idea is like sugar, basically very small polar things go and just become solutions. Like glucose is just a little molecule that goes in. Starch, we're going to talk more about starch. What is starch? Does anyone know off the top of your head what starch is? How is starch related to glucose?
Is the derived from glucose?
More.
The polymer of glucose?
Exactly, it's a polymer of glucose. Starch is a whole bunch of glucoses all connected together in big long chains. So it's polar, because each of the glucoses is polar, but it is big. It's a lot harder to get it to go into solution. You got to wait longer and mix it a long time. It eventually goes in, but it does scatter light. It looks kind of cloudy when you look at it. So when we are looking at the glucose solution versus the starch solution in these videos in a few moments, that's going to be one of the big differences, right? Both starch and glucose will dissolve, but because starch is this big wonky molecule, it actually scatters light when the light goes through it. I'll talk a little more about this stuff a little later, but let's just leave that. The third one, just for completeness, is a suspension. Here, the solute eventually settles. Think about if you have muddy water and you shake it up, it looks like muddy water, but then you come back in an hour and you've got silt at the bottom and clear water on the top, right? People have probably done that. Blood. Your blood is a suspension. If you take your blood and just put it in a glass, eventually the blood cells will all end up on the bottom and there'll just be clear plasma on the top, right? So a suspension is something where the solute stays up in there after you've shaken it, but after you let go, they all kind of come back and end up on the bottom. So does that make sense? Like a lot of medicines are formulated as a suspension. Like what do you need to make sure you do? If you have some medicine that's formulated as a suspension before you give it to somebody,
Shake it.
you gotta shake it. Because otherwise you're gonna have all the good stuff just sitting on the bottom and you're just gonna give them whatever this carrier is rather than the stuff they should be getting. So it's actually important to pay attention whether or not something's a suspension or not. If it's a suspension, you need to make sure it's shaken well before you actually kind of use it and distribute the solute through the thing. Okay. All right, so now, whoops, wait, where is it? Right here. Okay, chemistry review lab. This should be visible to you now. I gotta fix this. I'm not sure why I thought I fixed this, but it didn't get fixed. Doing the chemist. So This part here, submit PDF of Chemistry Review Lab write-up. I will publish it now. It's now live. This is going to be a place where you can submit your PDF when you're done with everything, but notice it's not due till February 8th. So we're going to do parts 2 and 3 today. We're going to do parts 1 on Tuesday. And then from then you'll have another week or two to finish writing it all up before you have to submit it. So
sorry. So part 1, 2, and 3 will be due on February the 8th, right? You said?
Correct.
Okay, good.
Yes. All right, so now we go into doing the chemistry review lab. So again, over the last few months, there's been 3 of us. There's Tina Christensen, who teaches one of the other sections, and Becky Brown, who teaches another section. We went into the lab and videotaped ourselves doing the different labs. So you would have something to work with here. Here you can see buffer systems. We're going to look at this one next time. For today, we are going to look at part 2, electrolytes, and part 3, carbohydrates. It says, for each of the 3 parts of the lab, you're going to record the data and observations in your lab notebook as if you were personally doing it. So, write down what you actually see, what we describe in front of us. You will also have combined class data for the different groups performing the buffer protocol. Don't worry about that yet. We're going to talk about that on Tuesday. It says make sure your discussion addresses the questions posed in the lab manual as you go through the various parts of the lab. Be sure you integrate your observations from part 2 and 3 into a cohesive discussion. Like I said, talking about how dissolving sugar versus dissolving salt, how is it similar, how is it different, and why? What's the underlying theoretical considerations explaining what you're seeing? Okay. Uh-oh. And now I am crashed. So hold on a few moments while I hopefully uncrash here. Dag nabbit. Please pardon our slight delay due to technical difficulties. We will resume with our regularly scheduled program in just a moment. Hopefully. All right.
I just want to make sure that I'm writing it exactly the way you want it to be written. So I put my name, my date. I'm going to put the chemistry review part at the top. And then right after that, do I write objectives and then start putting my notes in?
I would say that little part at the top has the objective, which is basically it's a demonstration. So
Okay.
It's kind of contained in there already.
Okay, thank you.
Yeah, later on, like for the jumping lab, you'll have to write a more formal objective where we actually have independent and dependent variables and stuff. But for this one, the objective is demonstrate some chemistry concepts. So, all right, I think this is gonna work. Please. All right, I'm going to stop sharing that. And I'm going to share this now. All right, so now we're going to do parts 2 and 3. Oh, you dork. Hold on. Parts 2 and 3 are hold on. To each other. It's just part 2 is dealing with electrolytes. All right, pause. Do the same this thing. Okay. I'm going to turn up the volume. And we're just going to watch this together. I'll probably pause it. But basically, this is available to you. So if you're not writing everything down right now, that's gonna be okay, but this, I want to kind of go through this with you right now, and this is going to be going through parts 2 and parts 3. And here we go. All right, so yeah, this is before I stopped cutting my hair too. I forgot. So now we're gonna do parts 2 and 3 of the chemistry review lab, and parts 2 and 3 are very similar to each other. It's just part 2 is dealing with electrolytes, and part 3 is going to do the same basic steps, but with carbohydrate, in this case, glucose. And just to kind of give you a sense of kind of this mechanics of putting the lab book together. You know, here we have data down from the last part of the lab, probably leave a little space to kind of write up, but then we're going to just have the instructions for part 2 here for electrolytes. Yeah, I'm doing this quick. In reality, you would do this a little more neatly and thought out. But basically, I have the instructions for the lab right here. So, and you can use a glue stick is better than tape, but tape works good too. And then I'm just going to follow. It's really important when you're doing the labs to follow it step by step. You know, we'll be reviewing, you know, here are the broad overview of how the lab goes, but even if you think you know what you're doing, you still got to slow down and read step by step, because there's often subtleties. And also, often we're taking the data from many different groups and we're averaging it all together. And you can't do that responsibly unless every group did the experiment in the exact same way. So it's really important, as much as you're gung-ho to just get data, to actually slow down, have at least like one person in your group who is the official kind of reader and gonna get more pedantic and, you know, so I'm gonna say part 1. What just happened? So, dag nabbit. So something. I do not know why this just choked. Gosh darn it. Yeah, normally this is not where the choke happens. Let's see. Let's try that again. All right. Here we go. Have at least like one person in your group who is the official kind of reader and gonna get more pedantic and, you know, so I'm gonna say part 1. Use a spatula to add small equal amounts, a large pinch of the dry electrolytes, sodium chloride, potassium chloride, and calcium chloride to 3 separate 50 ml beakers. So these are the little 50 ml beakers here. They're very cute. And we're gonna put a pinch. A pinch is basically kind of what it sounds like. There's a picture in your lab book showing a little bit. It should be enough that you can actually see it, but not so much that it's not going to dissolve in a little bit of water if it wants to dissolve. So we can come over here, and I've got each of these spatulas is labeled so I don't cross-contaminate between the different things. And I also probably want to make sure I mark what these are. So I'm going to use a pencil here, and I'm going to have some potassium chloride. I'm going to have a calcium chloride. And sodium chloride. Okay, that's another thing. In a lot of these labs, you're going to have very similar looking tubes or beakers, but with different stuff. And it's another place where you want to get kind of pedantic and be good about your record keeping and marking what's what so you don't get confused about things. So here, potassium chloride and a pinch, again, enough that you can see like the end of your fingernail, but not too much, because otherwise it's, if you try to dissolve a whole cup of sugar in a teacup or something, it's ridiculous. So I'm going to put all of these in, and then our camera will come and look at this a little more closely. I've got my calcium chloride, again a little pinch, and I'm going to put that in that one. And I'm going to get my sodium chloride and put that in here. And we're going to now, let's get a little more. All right. So I'm going to take these and put them like with a black background where I think it's going to be easier to see. And in each case, hopefully you'll be able to see there's this kind of these little white crystals at the base of each of them. Again, right here we have the sodium chloride, which is just ordinary table salt. This one is potassium chloride. This one is the calcium chloride. And this is where you're writing things in your lab book. When it says, describe the appearance of the dry electrolytes, you actually want to record that in your lab notebook. Use your pen and write down the words that describe, granular, white, fluffy, whatever words. This is kind of a qualitative description here. So, there's no right answer other than it should reflect what your observation is. And it needs to be something that you record while you're actually experiencing it. Memory is pretty imperfect. You don't want to go back and think, oh yeah, I think it was in a white powder or later on. That's not what your lab notebook is about. That's not what science is about. Empirical data is what you observe. So you need to record while you are observing it. So I guess the important thing about taking data is that you record what you actually see. And sometimes it's not what you expect. I should mention, obviously for this, you're not actually looking at it, but if you go back, we have a black background where I think it's going to be easier to see. Right, you can pause this thing. And right, so in each case, hopefully you'll be able to see there's this kind of these little white crystals at the base of each of them. Right, so you can go back and describe what it looks like. I mean, to the best. Obviously, it's not perfect because it's a video, but a lot of times what happens, especially again, because people kind of get this sense, this disconnection from the actual observation. They kind of write down what you think you're supposed to see, or look at it and use, it's white, it's kind of granular, it looks kind of like table salt, because it is, so that's still okay. You write it down. It's true and honest record of your work, not kind of what you would have seen in an ideal world or what you expect your teacher to write down. So. And I kind of I belabor that because people do what we call dry labbing sometimes, meaning they don't actually look at the stuff or do the stuff, but write down what they are pretty sure they should have seen. It's way easier to actually just write down what you think you should have seen rather than actually record what you do see. So don't do that. You kind of describe the things you do see. Now it says, after I've actually, I would write down, again, KCl. In fact, I'll look at it. It's, white, fine grains, etc. I'm not going to spend all the time doing it right now, but you would if you were here. Then I'm going to add water It says add 5 mils of distilled water to each beaker and then swirl the contents. To measure out the distilled water, I'm going to use a pipette. These pipettes are for much more accurate volume measurements. You always want to use one of these little pipetter attachments here, which basically sucks up. You don't want to ever use your mouth-to-mouth pipette because you might get something inside you don't want. These are a little confusing for people at first to understand. They come in different volumes. This one here is a 5 mil pipette, and it's designed to dispense volumes up to 5 milliliters. And the way it works is you would put it in the liquid you're trying to pipette, and the numbers actually kind of go upside down. They start at 4 at the bottom, and the 3 to 2 to 1, and 0 is up here. So the technique you would use is you actually suck up until you are at the 0. And that means now there is 5 mils in the pipette. And when I squirt water out, so this is just water, so this is actually safe. If I do that, this goes down to the 1. That means I have just dispensed 1 mil. I have now dispensed 2 mils, and I'm down to the 2. I've now dispensed 3 mils, I'm down to a 3. If I got it all out, I have just dispensed all 5 mils. So, because I need 5 mils inside each of these, I can just pull up to the 0. I'm going to quickly slap here. There's 5 mils there. Suck up again, up to the 0. 5 mils there. Suck up to the 0. So here we've got all of our electrolytes. And again, electrolytes, you should know what it means to be an electrolyte.
So I didn't actually, what do I mean by an electrolyte?
Something that can conduct.
Yeah, so basically any of these things that are ionically bonded, those salts, are electrolytes. By electrolytes, it means it's dissociating into ions, and conducting electricity. So I should probably just briefly, when we talk about ionically, bonded molecules, for instance, NaCl. Sometimes we call them salts, sometimes we call them electrolytes. Electrolytes also for ions, because electrical is involved with conducting electricity. So yeah, because the word salt, table salt, is one type of salt. KCl is also a salt. Salt is a more generic term than just NaCl. And electrolytes is also kind of a generic term for all of these dudes. So I should have said that earlier.
What do potassium chloride, sodium chloride, calcium chloride all have in common? And I've just mixed them with water. And now, kind of take a look. And here we're trying to decide, record the appearance. Can you see it still? Did it dissolve? Did it disappear? You know, I have to say, looking at these, they all pretty much look like they've dissolved. I don't really see the granules in there anymore. And then the next part is going to be testing for electrical continuity, basically. Oh, and I should mention, I'll say one of the interesting things. Remember I said write down your observations, but if they're really not consistent with what you expect, you should like maybe do a little bit of double checking that there's not a problem. It's not uncommon when we do this lab in person where somebody will mix salt with water and then write their description. Oh, it looks, it looks kind of like an oil slick. And they'll just write it down and move on. And it's like, all right, slow down. Wait a moment here. It's like, given your like life experience of putting salt in water, do you usually get an oil slick when you put salt in water? And it's like, no. So there's, usually what happens is, you know that we're going to be using oil later. If people have dirty beakers and they did not wash them well from the last lab, so somebody's adding water and salt into a beaker that's already got a smear of oil on the side. So you do want to, write your observations, but don't be like, be able to like think about, does this make sense? Do the reality check. In fact, we're going to talk about reality checks quite a bit as we continue on in this class, especially when you're doing calculations and stuff. Sometimes we call it a sanity check. Like, does this make any sense at all? I just weighed a canary and it looks like it weighs 356.2 pounds. All right, let's just write it down and move on. It's like, no, don't just write it down and move on. Something's obviously wrong. This canary doesn't weigh 300 pounds. There's got to be a problem here. So you do want to do kind of these sanity checks whenever you're doing this kind of work. Whether or not it can conduct electricity. And conducting electricity is basically, an electric current is moving charge. So for there to be an electric current, there has to be charge moving. In the wires, in the lighting in the room, it's electrons moving through the wires. In most biological systems, when you have electricity, the electric current is ions that are moving. So having ions, independent ions floating around in a solution typically is going to allow an electric current to flow. We can test that. I have this nifty little thing set up here. It's actually just a power supply, a little buzzer. This thing is a little buzzer, and wires that if I connect these two wires together, it completes the circuit, and you should be able to hear the buzzer. So wires are separate, no electrical current flowing from one wire to the other. I don't hear anything. I have the two copper wires touching. I hear it. So this is just a really simple way to see if something conducts electricity well. The air right now is not conducting electricity very well because I don't hear a buzzer there's no electric current flowing through the air from wire to wire when they're separated. I can take one of these solutions and again, these wires are not touching, but if I put the solution flooding, so both wires are underneath and submerged by the solution, I can see if I hear the buzzer going. And when you do this, you want to make sure that the wires have a lot of surface area in contact with the solution. If you just like barely dip their noses down, there's so little contact, you're not really going to get a good answer. But here, I'm going to take these. I'm going to put them in. This right here is the calcium chloride. Let's see what happens. All right, so on the air, I don't hear anything. In calcium chloride, I hear a buzz. I can rinse these little guys off with some distilled water. because I want to make sure I'm not kind of cross-contaminating stuff. As normally I would just have like a little beaker and edit that. So I'm cleaning these off. Now I'm going to go into my sodium chloride. And again, the same thing. Air, there's no electrical current. I'm going to submerge them in the sodium chloride. I don't know if you can hear that. You can hear the little buzz go. And then we can look for the continuity. We put the wires. They don't make a current in the air, but if they are submerged under the potassium chloride, I can hear a buzz. So again, I want to kind of in parallel so they're not touching. Want to make sure that there's a large surface area submerged, and sure enough. So that is that. The last part is it says pick any of those electrolytes, and now instead of dissolving them in water, put them in oil. So let's do that. Again, I'm going to take one of my little beakers. And it doesn't matter which electrolyte I pick. I'll pick salt because it's the one that table salt is the one I'm most familiar with in my life. I'll take some. Again, take a pinch. And I add it into my little beaker. And I don't need to label this because it's only one electrolyte. So this is the salt. If you take a look at it, it looks like salt. And now I'm going to take the mineral oil, and I can take 5 mils of mineral oil in the same basic way. Suck up to the 0. I now have 5 mils in my tube, and then dispense. That's going to be 5 mils of oil that are now in my beaker. And now I kind of mix it up. And I don't know if you can tell, if you can see the crystals. I don't see the crystals dissolving. I just see a bunch of crystals lying in the oil there. I don't know if it's possible. Does that show up there? And then I can check continuity, or the electrical continuity. Again, I take my wires and I'm holding them parallel. What are you going to expect? Are you going to expect we have salt in oil. Are we going to expect it to conduct electricity or not?
No.
No,
Why is that?
because the salt doesn't dissociate to mix in with the oil, or
Exactly. Since it's not dissociating, we're not going to have those free charge carriers. We're not going to have those cations and the anions floating around to be the electric current.
right.
Electricity requires the charges moving, and here the sodiums and the chlorides are still stuck together in their little crystals. Also, they're not touching. And I'm going to submerge them in this mineral oil salt solution. And make sure they're fully submerged. And now I don't hear anything. Right? Air, no buzz. Oil and sodium chloride, no buzz. Again, you can double check, fail safe, make sure the system's actually working. If I touch the wires, I totally hear a buzz. So the system is working. It's just I did not have an electric current when I was just testing in my solution. And that's more data I'd be recording in my lab notebook. And a little later on, I'll be spending more time kind of interpreting what does it all mean. But the first step is, while you're actually getting the data, is just to keep an accurate record of your observations. All right, so now we're on part 3. All right, so any questions so that was part 2. That was looking at the behavior of electrolytes.
in the polar and nonpolar solvents, and looking at both whether they dissolve, whether they conduct electricity. Any questions on that before we finish up with the sugar?
Yeah, I have a quick question. So I think before you were saying like sodium chloride, calcium chloride, they themselves we would call electrolytes.
Yeah, they're called electrolytes.
Okay. And so, if the solution produces a buzz, would we say what term would we use to describe that solution? Would the solution also be
We'd say it conducts electricity.
just that it conducts electricity? Okay.
Yeah. Yeah. There's different ways. You can say it has low electrical resistance or high electrical conductance. It'd be the same thing. All right. So we're gonna finish up here with part 3, which is just another few minutes here, because instead of 3 things, we're just looking at one thing. 3 of the chemistry review lab, which is basically the same thing as part 2, except instead of using an electrolyte, we're using a carbohydrate, in this case glucose. So I'm going to get my little 50 ml beaker, and I'm going to put a pinch of glucose in it. And I go find the glucose. I find the appropriate labeled glucose spatula. And come on, pinch of glucose. There's my pinch of glucose, my beaker, put it in. And again, you can kind of take a look and see there's a little granulated glucose crystals in there, which again you would record in your lab notebook. Then you're going to pull up 5 mils of distilled water. I'm going to add the 5 mils of distilled water. Agitate, give it a chance to dissolve if it's going to dissolve. And you can take a look, and it's worth, especially if you still have your other beakers at. Like I have a beaker of sodium chloride here. They both look pretty much the same. What does dissolve mean? The crystals have gone into solution. But then we're going to look at another thing. So here we looked at, does it dissolve or not, which we did with the electrolytes. We can now look at the electrical continuity. Again, I'm double checking. Yep, system is online. I'm going to hold my little wires parallel so they're not touching. And now I'm going to fully submerge them under this sugar water. And I don't hear anything. So that's telling me something about the electrical conduction of sugar water. And again, if you remember, if we were looking at the sodium chloride, again, it looks physically exactly the same. But if I put the wires in there, it's a very different response. So like in your discussion, parts 2 and part 3, even though they're kind of separate parts, in your discussion, you should probably kind of integrate your observations for both the electrolytes and carbohydrates under the same conditions, and how they are similar and how they're different and why that makes sense. The last part of the carbohydrate part is just looking at the optical difference of what sugar water glucose solution looks like compared to starch solution. In starch, which is just a whole bunch of glucoses bonded together in big long chains, but when they go, and it will go into solution, but becomes a colloid. And it's worth taking a look here. This is the sugar water, a true solution, which by definition is not going to scatter light, is going to basically be transparent. Whereas when you have a starch solution, I don't know if you can kind of notice it here, a colloid does scatter light, which basically means it's kind of cloudy or milky looking. And hopefully that, part of, this is a demonstration lab, trying to get you to just have a more personal sense of like, oh yeah, I see that glucose dissolves into a transparent solution, whereas the starch is dissolving as well, but it's creating a different kind of mixture, this colloid, which you can physically see as being different. So, all right, so that, and I do know that it's not nearly as compelling and thrilling to watch your teachers do things than instead of doing them yourself. So, I mean, unfortunately, there are going to be parts. There are going to be parts of our lab where we will be. You'll do stuff on your own at home. But for a lot of times, we'll also be doing things like this, where you get to at least you'll get the main points, take home messages. You'll see things. We're connecting the ideas of lecture into actual physical observation, which I think will help you kind of really own the material at a deeper level. And at the same time, I do acknowledge, this is not as cool as going in and mixing stuff up in person. So, but I think it's better than getting COVID. So, any questions about all of this labby stuff?
I do have one question. I know I keep asking you about it, but I just wanted to make sure. So since we're starting with part 2, should our first page be part 2, or should we leave a page blank for part 1?
I would start with part 2. Yeah, no, that's a good question. I think just for the basic flow, that makes sense too.
I have, and then, sorry. So like, for example, like in part 3, question 3, it says draw a structure of glucose. You want us to still do every step, right?
Yeah, so in fact, here, it's not a bad, share screen. So here is a glucose. So glucose has different forms, right? There's this kind of linear form, but when you put it in water, it zips around into a circle. It's actually more like a chair, but it's basically, you can think of it as a circle. So when you're drawing your glucose, you should draw it, like this. This is a glucose. It's got 6 carbons, but they're not all in a ring. One of the part of the ring is an oxygen here. And then it says to show how water makes a hydrogen bond. So if you're doing that, I would say remember that while overall this thing stays intact and overall this thing is neutral, it has parts of it that have charge imbalance. Like there's the lone pair of electrons on the oxygen, so this is going to be a little more negative. Where you have a hydroxyl, an O actually attached to an H, the oxygen's going to be more negative and the H is going to be more positive. So you have to think about this sugar molecule as this thing that has sections that have more positive, more negative. Let me, so more negative, more positive. And then think about adding water into it. And again, a water molecule is just going to be an H2O. And you're going to connect the H2O with solid lines because that's the way that you denote that there's a covalent bond. But then you're going to also have your connection to the water as a hydrogen bond. And remember that a hydrogen bond is denoted in this more kind of temporary way, as this kind of dotted line, right? So, like, okay, does that make sense? Like the hydrogen bond is not a solid line. It's kind of the dotted line showing that it's a kind of one of these kind of more temporary attachments, which can break apart as well more easily. So that's kind of what you're doing in that section when you're trying to draw the sugar molecule, and then really think about what does it mean to dissolve. It means that the water molecules, the polar water molecules, are having these polar interactions, plus, positive negative attractions between the positive or negative parts of the water molecule and the complementary part of the sugar molecule. And that's why sugar is dissolving in the first place. That makes sense? The other part in there, just for completeness, if I have NaCl and it's a solid and it dissociates, what is it going to break apart into?
Na plus and Cl minus.
And both of these are aqueous. I'm trying to draw. So this is the dissociation of sodium chloride. Let's look at this one. Calcium chloride. What happens to this puppy when you put it into water? This is one of the ones we were using.
Calcium 2 plus and 2 Cl minuses.
Exactly. Remember, calcium also ionizes, but it loses 2 electrons. It has 2 electrons in its valence shell. So this is going to be calcium 2 plus. And then each of these chlorides is going to be a chloride ion, right? So it's going to be a Cl. You could do it like this, or you could say 2 Cl minuses. But it's going to actually, how many individual ions will we get floating around in the water for every single calcium chloride? I saw Tim's got his fingers up there. 3, exactly. So it's very different. Like when you put a sodium chloride in the water, you get 2 ions. When you put a calcium chloride in the water, you get 3 ions. And again, this is all going to be relevant when we start talking about osmolarity and stuff like that. You actually need to pay attention to this kind of stuff. How many particles do you get in the solution? Depending, and it's going to depend on how many ions this thing dissociates into. And we're going to talk about that in more detail on Tuesday. But when we start talking about osmolarity, it's going to matter a lot whether or not things dissociate or not, and how many pieces they dissociate into. What else? Any other comments or questions about this?
Just like the part that we just did, as well as drawing the structure of glucose, that just goes in with like in order in the observation area.
Yeah, that'd be
Okay.
Uh-huh.
Thank you.
And then in the discussion, you'll kind of really bring in the ideas of why things look like they do and everything. What else? So, we'll probably leave it here. What we're gonna do on Tuesday is the part 1, which is the buffers when looking at pH. And we are also, I think what we'll probably do is do a test. So Proctorio is, I'm sure a lot of you have experienced Proctorio by this point if you've been doing online classes. It's this kind of way to do a more secure online test, but it's kind of more involved. You have to make sure your computer is set up right and everything. So we are going to finish the lab, and we'll also do a little practice. It won't be worth any points. It'll be more just to make sure your computer works and you understand how Proctorio works, and so we don't run into any hiccups when we have the real exam. So we'll do that next week as well, as part of the lab time, just to kind of get that out of the way. And we'll talk about a few other things. We'll also talk about the jumping lab, because for next Thursday, you are going to have an objective and a hypothesis for your jumping lab. But don't worry about that until Tuesday. We'll talk more explicitly about what the expectations are. So next Tuesday, we'll finish this chemistry lab, kind of introduce the jumping lab, and we'll also do a little bit of the getting things in ready for testing with Proctorio. Otherwise, though, I think we're gonna end a little early today. So if you have any questions about anything, please stay online. I'll be here till 1:30. Otherwise, have a good weekend.
Thanks, you too.
Thank you.
All right.
Thank you, Dr. Egert.
Thank you.
Thank you. Those lab videos are going to be in our modules?
They are in your module already.
Perfect. Thank you.
Sure.
Is it kind of just standard operating procedure to sit in class, go through the lab with you in case we do run into any questions, but then to probably go back into the modules and go through it a bit more thoroughly to, put our data down, that kind of stuff?
It'll be different. Different ones will run differently. So I would say there is no, in some of the ones we'll do more, like when we all went into the breakout rooms to talk about the variables. We'll have other labs where we'll have more time where we're in meetings with each other. Some of the other labs will be just setting up what you do, but you're actually going to do it at home with stuff you find in the kitchen. So it's going to be a bit of a smorgasbord, I'd say.
Sounds good. For today's lab, I started to try and jot stuff down as we went through it and realized I think I'd be better off just going back, and that way I can stop the video and take a good gander before I write down my analysis.
Yeah, absolutely. I think that makes total sense.
Thank you very much.
Sure.
Enjoy your weekend, Professor. Thanks.
You as well.
I just had a question about some of the stuff on the lab. Like it's not labeled number 2 or 2A. It's like one of them is how do water and oil differ in their relationship to electrolytes. Is that just to kind of help us get
getting you more to think? That's getting you to think. That's something that's going to come up in your discussion.
Okay, perfect.
Right. In your discussion, you'll elaborate on that. But I think it's just in the moment, it's there to kind of tickle you a little bit.
Perfect. All right. Thank you.
Sure.
And I have a question about, I guess, like 1B. So it's asking what happens when we test the electrolyte solutions with the electrical continuity tester. So for the observations, would we just say if we heard a beep or didn't hear a beep?
You can say,
and then in the discussion, we would go into what that means to hear a beep or not hear a beep.
Or, I mean, I would say it makes sense even because we know that the beep means it conducts electricity. So you might want to just write, heard buzz, conducts electricity. And later on in the discussion, you'll talk about why it makes sense that it conducts electricity based on
the solution conducts electricity. Okay, perfect.
Sure.
Thank you.
You're welcome.